Okay, hello everybody. Sorry, back. Okay, the last slide of Mr. Eyes was about Syngenta, a Swiss enterprise. I hope I will give a better image of Switzerland right now. Okay, the title of my talk is The Importance of Polymorphism in the Face of Global Change. I would like first to thank my collaborators, because I'm not working alone, as you can imagine. I'm working with several. Okay, I can work. I prefer. Okay, I can work with several people on population dynamics, a group in Norway, molecular evolution as well at the University of Lausanne, Nicolas Salama. The genetics, all results I will present on the genetic parts have been produced by Annelise Ducrest and Céline Simon. With Jérôme Goudet, who is here in the audience, we are working on phylogeography and genomics, ecophysiology, behavioral ecology, and recently special topics about sleep as well. I will not have the time to present everything, but some parts of this work. Okay, the last times I was a bit thinking of to present in one slide how I think world is. It's not very easy. And I came with this slide. I hope you will agree with me. Okay, thus you can see that there is a lot of variation in habitat and changes in habitat due to global change, as we can see, which become more dry. And animals evolve ways to deal with all these stressful factors, and there are genetic adaptations which affect the physiology and the behavior of the animals. And a key issue is about the maintenance of diversity, including genetic diversity, and we can observe at the moment microevolution that's due to global change. You can have several responses to global change, as you already heard this morning. For instance, if you are not happy where you are, you might just disperse to find a better place, like everybody did today when Sevilla. It's better than in Norway, apparently. You can have phenotypic plasticity, but today I will talk more about potential genetic solutions to this global change, because the response is as quoted by a paper by GNAP. As you can read here, there are limits to plasticity responses, and they are unlikely to provide long-term solutions for challenges faced by population experiencing directional selection on the long term or mid-term. Okay, I will talk more today about a special case of, I'm working myself for a couple of years already on color polymorphism. And this color polymorphism is quite interesting because the different morphs you can find in a population, for instance, in this lizard population, the morphs are under very strong genetic control. Thus, if you are yellow or red, it's not because you eat some yellow food or reddish food, but because you have the necessary genes to produce these color variants. And these traits, there are more and more studies at the moment showing that these traits are associated with many physiological adaptations or behavioral adaptations. Thus, you can consider these morphs as phenotypic markers of adaptations. Most color polymorphisms are based on one type of pigment, which is the melanin. It's widespread in all vertebrates. It's the most common pigment, in fact. And here the examples I show you is color polymorphic species. It means that within a given population, individuals of the same sex can display 1, 2, or 3, 4 different variants. But you have to keep in mind that many animals, including human, by the way, there is continuous variation in the degree of melanin-based color. That's what the things I will talk about color polymorphism apply as well to species in which there is continuous variation in the degree of melanin-based color. Either black coloration due to eumelanin or reddish coloration, pheomelanin. I began to be interested in the link between color polymorphism or melanin-based color and global change. And just for fun, more or less, I was looking in a book on our species, and I just looked where each species are located on the planet and what is the degree of reddishness. And what you can see here, it's quite clear, on the northern hemisphere, but not on the southern. On the northern hemisphere, the more you go to the pole, the more gray or non-reddish the species of owls are. This might suggest that the degree of reddishness is adapted to quite warm climates around the equator, and gray species, non-pheomelanic, non-reddish species, might be better adapted to climates in the northern parts of Europe. And in fact, I made this analysis at the same time as I received this paper to review. And I thought, wow, that's really great. This is the study of Karel and Bromer. What they found in Finland, they used data collected by ornithologists, amateur ornithologists. And what they show is that there is a huge increase in the proportion of reddish individuals in Finland during the last century. And thus it suggests that since climate it's more warm at the moment, if to be reddish is an adaptation to a warm climate, the conditions becomes better for reddish birds in Finland and right now, explaining the increase in the proportion. And what is very important, it means that for a polymorphic species, you have gray and red individuals. If there is suddenly a change in the conditions, the probability that this species will persist in the face of global change is higher because you have one of the two types which might better cope with the new conditions. Interestingly, Galeotti found exactly the same pattern with another species of bird, the Scops owl, in which you have the gray and the reddish morph. And in Italy, you can see that right now, the proportion of reddish birds is higher. Myself, when I was in the museum in Geneva, I was just looking very quickly at the skins of tawny owls. And 10 years ago, I already noticed, just by pulling the drawers, I said, wow, very gray, wow, very reddish. And when I was looking at the dates, completely different dates. This is maybe a quite general phenomenon. That's just what I said. The gray owl, it's gray, it's monomorphic. The tawny owl, you have two morphs. And I could well imagine that this type of species, the polymorphic species, might better cope with global change than this one because they have more variation in genes, which might be useful to adapt to the new prevailing conditions. Okay, these are observations, and I will not say right now that I have the solution, not at all. But I will present the research procedure that I'm doing for already a couple of years. And why, my main question is why melanin-based color is associated with global change or other habitat features. And this is very important since I said at the beginning of the talk that melanin pigments are widespread in all vertebrates. And the genetic system underlying the production of melanin pigments is very highly conserved, meaning that the genetic system you have in dogs or in birds or in lizards is more or less the same as in fish or amphibians or reptiles, or laboratory mice, by the way. And I came with an idea a couple of years ago because I was working with owls, and I always found, I will show you results, very strange results. And I was wondering, why do I find these results? At the start, I did not believe my own results. But after a while, when you get new results, okay, there should be something. And we opened a biochemical book, and because the genetics of melanogenesis is very well known, and we were struck by one system in this book. This is the melanocortin system. The reason why we were particularly interested in this system is because one gene called the pro-melanocortin gene produces the MSH, alpha MSH, which triggers the production of eumelanin pigments, black pigments. And there is an antagonist, agouti, which triggers the production of pheomelanin, reddish pigments. And these hormones not only bind to the receptor responsible for melanogenesis, but to other receptors, which affect many different physiological processes and behavioral processes. And we did a review of pharmacological and genetic literature, which was very complex, took years. And we reviewed all these papers who use different technologies like knockout mice, or mice which overexpressed some genes, or animals in which you inject the hormones, or in which you inject antagonists, so that you don't have the effect of the hormone, you prevent the effects. Okay, this is a summary, quite complex. The idea is not to go into all details. The key thing is that, as you can see at the top over there, the MC1R receptor, the melanocortin-1 receptor, this is the receptor on which the MSH binds, and you produce the black pigments. But as you can see, the MSH or ACTH, which is also a melanocortin, binds to 4 other receptors. And these 4 receptors trigger many physiological processes, like the response to stress, corticosterone, the MC2R, very important. MC4, for instance, in human, is associated with obesity. There's energetical processes, immunity, aggressiveness. If you inject MSH to mice, they become very aggressive. Or sexual behavior, because melanocortins affect sexual hormones as well. Many behaviors and physiological processes. Thus, what you expect is genetic or phenotypic correlations between all these traits. I mean, if an animal is black, it has some specific behavior or physiological physiology compared to lighter colored animals. Okay, just to sum up the genetic, you have the MC1R, the POMC gene produces melanocortin hormones, which bind to the MC1R, and you get a black mice. If you have the agouti, as well some new studies for the specialists, agouti-related protein might play a role as well. Binds to the MC1R, it prevents the production of black pigments, and you become reddish. Okay, this is a review of what you would expect if you increase the level of melanocortins in animals via overexpression of the genes or injection of melanocortins, all these traits. And for instance, you can see here, you better resist, I don't remember what it is exactly, but you better, if you food deprive animals, if you have more melanocortins, you better resist lack of food. In the opposite, if you have more melanocortins, you have a lower appetite. And we tested this, for instance, these predictions in the barn owl, and this is exactly what we found. If we food deprive animals, blacker animals better resist lack of food, and they have a lower appetite. Thus, they better withstand food deprivation or habitats where there is less food. This might be one adaptation. In general, melanism is found to be associated with resistance to stressful factors. And that's why there might be a link with global change. Okay, I will present you a couple of new results, some just published or very soon, and unpublished results. Even was doing analysis yesterday evening because we just got the data. There's preliminary analysis. And we did these studies with 2 polymorphic species. The tawny owl, you can see, is very cute birds. You can see 2 morphs, the siblings. And the barn owl in Switzerland, the studies were performed. And the barn owl varies with respect to reddishness, from white to dark reddish for melanin pigments, and black spots. Maybe you see them, more or less large spots, eumelanin pigments. Just to show you a summary of the studies for 15 years about, or 10 years here. This is the change in the population, in the degree of melanism. As I could already see in the field myself by measuring the birds, say, wow, they are blacker than a couple of years ago. And when we plotted the data, indeed, they become blacker. Thus you have the males below, the females above, and nestlings and adults. The nice thing is that the nestlings already express these melanin-based traits. And this change in the population is due to survival. Selection is on survival. It's not on fecundity, production of chicks, etc. It's only due to survival, and only in the first year of life. After the first year, selection is no more very strong on survival. And as well, population dynamics of the barn owl is explained by variation in survival in the first year, not at the survival on adults. Interestingly, by doing quantitative genetics animal model, we could show that the breeding value, thus the additive genetic variance, the change is due, just in the population, birds become blacker. The change is due to a change on genes located on the autosomes, not on the sex chromosome, because this trait is due to genes on sex chromosome and autosomes, but selection is acting on genes on the autosomes. Okay, just to show you one experiment where the link between color and other physiological traits is very, very strong with the glucocorticoids, the regulation of glucocorticoids. That's the hormone which deals with stress, whatever the kind of stress, immunity, lack of food, social stress, etc. If you are stressed, you increase the level of corticosterone. And this is good because you can flee. You know, if I come with you a very nasty, you will be scared. The level of corticosterone will increase and you will flee. But to keep this level very high is not very good for you because it depresses, for instance, the immune system. And based on the idea that the melanocortins binds to the MC1R, which triggers eumelanin-based color, MC2, responsible for the regulation of corticosterone, we expected a link between color and corticosterone. And this is exactly what we found. This is one experiment where we stressed birds. The chicks were raised by foster parents, so that we compared the glucocorticoid response of the chicks raised by foster parents with the color of the biological parents. And you can see that the chicks born from black mothers have a lower glucocorticoid response, and thus they suffer less from glucocorticoids. This depresses less growth and immunity. Okay, so far this was more or less correlative studies, and now we dwell into the genetic system. And we just got some new results on gene expression. We're looking at polymorphism as well, but we just finished the work on the gene expression. And thus, our idea was to see whether the degree of melanin-based color is associated with the expression level of the genes belonging to the melanocortin system. This is a key assumption. It doesn't show that the melanocortin system is responsible for everything we find, but at least this is a necessary prediction to test. Okay, here I show you the part of the cascade leading to eumelanogenesis, production of melanin pigments. You have the POMC gene, and there is something very important. The POMC gene produces a very big hormone, which is not biologically active. You have to transform this hormone to get biologically active peptides. And you have two convertases, proteases, enzymes, which cleave the prohormone, the PC1 and PC2. And what you can see here is that the level of expression of POMC is indeed significantly associated with the color, and as well the expression of the convertase, which is associated with the color. And thus it fits with the prediction of an association between gene expression and the coloration, in the feathers, by the way. We collected the feathers, not in the brain. That's something else we should do, but we cannot kill animals like this, like mice, to do these experiments. Thus we do the best we can. And we did more or less the same study in a better way, I would say, in the barn owl. We just got the results, just drew the graphs in the plane. We as well found an association with the POMC gene and the degree of reddishness. In fact, we measured the quantity of melanin pigments in the feathers. Thus, this is the concentration of pheomelanin pigments in the feathers. The PC2, the second convertases, is as well associated, sorry. And what is very surprising, we did not find anything with AZIP, which is the antagonist, but we found something with AGRP, agouti-related protein. And I think this result is very important because agouti-related protein is in the brain, which is not the case with AZIP. And everything is going on in the brain, of course, to get all this association between physiology, behavior, and color. And if the expression of agouti-related protein is correlated between what's going on in the feathers in the brain, this could explain many of our results we observe in nature in natural conditions, and the results that they found with the tony owl in Finland. Okay, to finish, maybe the melanocortin system, my account for the covariation between Mendeleev scalar and adaptation to stress. And maybe it might explain these results. And what the key thing is that this system is conserved in vertebrates. That's what we find with barn owls, tony owls, is as important as what we find with laboratory mice. That's the same system, that we can work in natural conditions. And of course, I thank you for your attention.